yeah. So I
Perfect.
would like to first to present Rasha very quickly.
Mhm.
It's a pleasure to introduce Dr. Rasha. She is my sister, science sister. Yes, we have the same supervisor. My
Yeah.
case was like last century. Yeah,
Not when you say it that way.
yeah, it's the same guy, same guy.
Yeah.
So it's very nice to have you here. We
Thank
have,
you.
yeah, talking for a long time about you giving this presentation. You can always give a talk with your past results from Ken Story. Uh, in another occasion will be very good. And so, and I would like to thank you everyone who is attending, and we hope we have fun. Yeah.
Awesome. Thank you so much, Marcelo. So thank you so much for inviting me and giving me the opportunity to present my work from my graduate school up until now. I'm very excited to present my work to you guys. And if you guys have any questions, please stop me whenever. Send me emails. You have my phone number. I'm on the group chat. So like whatever you have a question, just let me know. And I'm more than happy to have a discussion, answer questions, and what have you. So today,
Yeah, you can put in full display.
oh, sorry, like that. That's what I was asking. Like I was in, because I have like two screens. So can you see the full display now?
No.
Oh, 1 second. Because right now I see both of my screens. Sorry. This window. Can you guys see it now in full screen?
No, it's the same as
Okay.
before.
How about now? Because right now I switched the.
Not
Okay.
yet. Not yet.
Okay. If that, I can just connect the second screen, and then that way it'll be much easier. Yeah. Let me do that just in case. We have other people have other things to do. Oops. Hello. Can you guys still hear me?
Yeah.
Fantastic. Okay. So let me share my screen again.
Oh,
Window.
we have Max right now.
Wonderful. Wait, where did my screen go? Oh. Okay, now I don't see any of my presentation.
We have an Argentinian with us not right now.
Yay! Awesome. All right, so can you guys see my screen now? Full screen?
Yeah,
Go from the beginning.
but it's still not in full display.
Yeah. How about now? Now it should be good. I have like one screen. It's just on my laptop. Let me know whenever it loads, and I'm happy to start.
Yeah, it's
Okay.
still not in full display.
Do you see like the presenter view, or do you see like is it just like still loading?
I see the small slides on the left.
Oh, okay. So it's probably still loading because hold on. Because if I do it full screen, then I won't be able to like click on
Okay,
any.
you do the best you can.
Okay. Let me do this and then see. How about now? Do you hold on?
Yeah, it's better. Yeah,
Let's
as
go.
long as we can see
Full.
the
Yeah.
understand the data, that'll be okay.
Okay. Let
Because
me go
sometimes
back
it's
to
too
this.
small. But for me, it's okay.
Let me try to like remove things like I don't know how can anybody help to what was it? Does anybody know if I do it full screen and then share it, like if that's gonna help this? Because like to me, like I have it as full screen and it is sharing, but I'm not sure how like
Uh,
what others see it.
I'm not the best person to answer this question. I am a Neanderthal.
I should be like familiar with this, but I usually use Zoom. So like I'm not sure with Google Meets if there's something. Um
Yeah, so what I would suggest to you is because you're not getting there, in
Yeah.
theory, you can select the screen that you're presenting, but if it's not, just hide
The
the
other
bar
thing? Yeah.
and remove the
Oh,
add
yes.
notes.
Okay. There you go.
So if you also hide
Perfect.
Yeah.
the
Great.
bars from
Yeah.
the top,
Okay.
that one,
Oh, I should have been. Okay. So, let me see. How do I remove this?
just click in this arrow on the right hand side.
Yeah. Always ribbons. So, tab only. There you go.
Yeah.
Okay.
Uh,
Perfect. Can you guys see it now?
yes.
Amazing.
Yeah.
Okay.
Yeah. It's perfect. Perfect.
All right. Sorry for the technical error issues.
No, it's
Um,
okay.
So today I'll be talking about my journey of how I started from Dr. Ken Story's lab in Canada, Toronto, all the way to how where I got to be now at Dr. Shannon Tessie's lab at Harvard Medical School and the Massachusetts General Hospital. So during my undergrad, I went to the University of Ottawa and I studied biochemistry with concentration in immunology and microbiology, and I was really interested in studying aging, so I studied gerontology. And then in 2014, as soon as I graduated, I went to Carleton University in Ottawa and did my master's and stayed for my PhD, specializing in biochemistry and molecular biology with Dr. Ken Story. And then in 2020 to 2023, the middle of a global pandemic, I moved to Toronto to the McEwen Stem Cell Institute in order to study stem cells and regenerative medicine with Dr. Michael Laflamme. And I'm currently at the Center for Engineering and Medicine and Surgery at Harvard Medical School. And I'm focusing on organ preservation. And I'm thinking of staying here for another like a year or two before hopefully finding a faculty position in regenerative medicine. So as we all know, this is the famous Ken Story, wonderful PI that really I wouldn't be here without all of his help, his knowledge, and his laboratory. So in my graduate training, we focus on understanding the molecular biology of how certain animals in nature can tolerate extreme conditions that are extremely lethal to humans. And I was interested in studying the molecular mechanisms that allow these animals to cope with such extreme conditions. So going back to the central dogma of stress biology, we know that during stress, like during development, we have DNA that turns into RNA, it turns into protein. But then during the stress response, you kind of have this like back and forth interaction between them. You have regulation at all sorts of biological levels in order to create a coordinated response that will allow the cell to survive. However, at some conditions, especially in non-stress tolerant mammalian models, this regulation does not happen in a coordinated manner, therefore resulting in pathologies. So first, I will discuss a few of the work that I did at Dr. Ken Story's lab, and then kind of like merge that to my research with stem cells. So I'm sure like all of you guys are familiar with the freeze-tolerant wood frog. This is the animal that I did my master's and PhD in, but I've also had the opportunity to work with other unique animal models. So in winter, this animal goes, and as soon as the fall happens, it starts eating a lot and then accumulates large levels of glycogen in its liver to use as both cryoprotectant and fuel source during the freezing process. When the temperatures gets colder, this frog goes under layers of leaf and litter in the forest floor and finds a place to hide basically for the duration of the winter. And as soon as ice crystals from the environment touch its skin, it starts the ice nucleation process. And as you can see here from Dr. Rubinsky's work with Dr. Ken Story in 1994, that when you put these frozen wood frogs under an MRI, you can actually see the ice propagation from the outside very uniformly to the inside of the body. And subsequently, when you put these frozen frogs into the MRI, they don't thaw uniformly. The organs that have high levels of glucose that has been secreted before the full initiation of the freezing process as a cryoprotectant will cause the different organs to thaw at different times. So for example, whilst the liver, the heart, and the brain have the most glucose, they thaw faster versus skeletal muscle, which has the least glucose, it will thaw later. So I'm sure you guys are familiar with this, but basically during the freezing process, we have a timeline. So we have glycogenolysis that occurs during the first hours of the freezing process in order to distribute glucose throughout the body. And then you have the for longer extended periods, you have the heart stops beating, there's no breathing, there's cessation of mobility, and the frog stays like that for a duration of its freezing, which could be up from days up to weeks up to months at a time. And as soon as you put this frog at a temperature that is a little bit higher than its body temperature, then it will start the thawing process, reversing all these regulatory mechanisms in order to resume full function. And we can see this frog within even as early as 2 hours post-thaw start stretching, and within 8 hours to resume almost full function without experiencing any measurable damage. So with that being said, these animal models provide a unique opportunity for us to study how these molecular mechanisms are protecting the animals, so that we can use therapeutics and genome engineering in order to improve the preservation of transplantable cells, tissues, organs, and what have you. So again, this is a video of the freeze-thawn wood frog, which I think because I'm in the presenter view, it's not allowing me to see. But basically, this was just like a video to show that a frozen frog, how it slowly comes back to life. So in doing this process, the wood frog tolerates freezing, hypoglycemia, dehydration, and anoxia that it can also tolerate independently of being frozen, which gives us a multifactorial animal model to dissect the specific stresses associated with freezing independently of each other in order to see their summative effect during the freezing process. But the one thing that allows this process to occur is a coordinated response of metabolic rate depression, which reduces overall metabolism and allocates a finite amount of energy available only to pro-survival processes. That is, for example, apoptosis, growth, development, all of those are arrested, versus antioxidant mechanisms, defense mechanisms are basically upregulated in order to protect the animal during the freezing process and also during early hours of recovery. So going back to the central dogma of biology, I've done a couple of studies at different levels that I will be highlighting today. So at the transcriptional levels, I studied several transcription factors in different tissues, and basically saw how the different stresses associated in the wood frog differentially regulates their expression, localization, and downstream activity. Today, for this talk, I'll be focusing on carb loading, and this is basically studying the transcription factor ChREBP and its regulation transcriptional network in wood frogs during the freezing process in liver and skeletal muscle. So basically, ChREB is a transcription factor regulated by phosphorylation, and obviously, like the different kinases and phosphatases that mediate the phosphorylation and dephosphorylation events are regulated by stress responses within the cell. So as soon as ChREBP is phosphorylated, it goes into the nucleus and binds to the promoter of specific genes, causing their transcription. So in here, what my study showed is that we see that the protein levels of ChREBP is highly upregulated in the liver, but is downregulated in the muscle, which makes sense. Liver is a highly metabolically active tissue versus the skeletal muscle is very dormant. So comparatively, comparing like liver and muscle is interesting because you can see and compare the contrast between active and non-active tissues. So when we measured using Western blot, when we measured the protein levels, we saw that the protein levels are highly upregulated. The mRNA levels are highly upregulated in the liver while staying consistent in the muscle, as well as the relative nuclear localization, which tells you that this transcription factor is actually active, is upregulated in the liver but not in the skeletal muscle. Then we measure the DNA binding activity, and this is basically you're taking the nuclear fraction and then doing an ELISA-based assay where you try to see the binding activity of the transcription factor of interest to its consensus sequence that's already adhered to the bottom of the ELISA place. So we can see that the DNA binding activity is actually upregulated in the liver and downregulated in the skeletal muscle, which kind of makes sense because, again, liver is a metabolically active tissue. So for the, I'll be focusing on SCD, PK, and FAST, which are the immediate downstream targets of this transcription factor involved in fatty acid synthesis, as well as catalyzing some of the glycolytic metabolism and de novo fatty acid synthesis. So what was interesting is that whenever I measured the transcript levels of these three genes, I saw a significant upregulation in the liver, but not in the skeletal muscle. The skeletal muscle only pyruvate kinase was upregulated, which was interesting because we know that from previous studies in the lab, we know that fast fatty acid synthase at the protein level, it's actually downregulated during freezing in the wood frog liver. So this contrast made me think, is this regulated? Is this pathway regulated at different biological levels? And we know that with the wood frog, during freezing, all the metabolism is reduced. So anything that it needs immediately after thawing, it would have to upregulate during in the pre-freezing period and basically keep it in a dormant state in order to use it as soon as it needs it during the recovery. And with, for example, fatty acid synthase, it would need it because it would need to repair put any potential damage to the membranes. So because there was a discrepancy between the mRNA levels and the total protein levels of fatty acid synthase, I went and dived deeper into more studies that we've done previously in the lab. And interestingly enough, the microRNA, so microRNA 21 and microRNA 16s, would regulate the translation of this mRNA, are actually upregulated in the liver during freezing. So what this tells me is that even though the transcription factor itself seems to be active and doing the activity, the downstream factors involved in in the other processes, the downstream genes, are regulated and only activated at such time where the wood frog needs them, such as during early hours of recovery. And this is very, very important because had we not looked into the whole pathway, then we wouldn't have known this complex regulatory mechanisms. Just looking at the mRNA levels would have told us that these genes are highly upregulated, these proteins are active, and it would have painted a different picture in the in terms of us understanding the molecular mechanisms of freeze tolerance. Another couple of studies that I've done, again, going back to the central dogma, is at the translational level. And we know that with translation is an extremely energy expensive process. So because of that, most of the animals that actually endure such stresses in winter downregulate their protein translation in order to conserve the finite amount of energy available until full recovery. So for this, I'll be focusing on the naked mole rat, which is a notorious model for being cancer resistance, for being hypoxia tolerance, and being able to tolerate stresses that are not tolerated in terms of hypoxia in other mammalian models. So for this, we use Luminex technology in order to understand the post-translational modifications of several markers involved in the translational machinery. So using this, we found that there's a bunch of different phosphorylation events that's happening with respect to the mTOR signaling pathway, as well as the translational machinery, that in summation will conclude that cap-dependent, sorry, to move this so you can see some of these results. We see that there's differential regulation of these phosphorylations that in summation would mean that there is an overall decrease in cap-dependent protein translation in some tissues, while other tissues have cap-independent protein synthesis. So why is this important? Majority of our genes are translated using the cap-dependent pathway. However, certain stress-responsive genes, so for example, antioxidant, heat shock proteins, and what have you, have this iris element at their close to their promoter, so it allows the ribosome to kind of skip that finding that cap and basically initiate translation in a cap independent pathway, and this allows the cell to basically discriminate between which genes to upregulate and which genes not to upregulate under conditions of metabolic rate depression. So I wanted to see then, okay, I've done all of these studies and I've done a few other studies as well, but for the sake of time, I just wanted to focus on a few more. So after finishing my PhD, I wanted to see that would recapitulating these pro-survival responses in nature improve post-transplantation outcomes. And for this, I joined Dr. Michael Laflamme's laboratory at the McEwen Stem Cell Institute at the University Health Network and the University of Toronto in Canada. In 2020, and Dr. Michael Laflamme's work focuses on using human pluripotent stem cell-derived cardiomyocytes in order to replace damaged tissue post-myocardial infarction in vivo. So basically, after myocardial infarction, after you have like a myocardial infarction event, you have the death of the downstream targets because you don't have any blood or nutrients or oxygen delivery to the tissue. So a part of the left ventricle dies. And right now, we do have medications that can reduce the severity of the effect and to delay disease progression. However, we don't have any curative effect. So for now, we have two remedies that we can do. We can have heart transplantation, which is quote unquote like the only available curative therapy. But unfortunately, we don't have enough donor hearts to give to every recipient that is needed. And as a result, many patients go without this life-saving treatments and unfortunately die. One other way to circumvent this is to use cell-based therapies and to repair the damaged tissue in vivo and to use stem cell-derived cardiomyocytes in order to remuscularize the infarct scar in vivo. And this can be done using single cells, cardiac patches, and engineered microtissues. My work focused on using single cells in order to remuscularize the tissue, but some research has been focused on using cardiac patches. And actually in Germany right now, we have the cardiac patches going into clinical studies because they don't have the same side effects that we see when we try to remuscularize the infarct scar with the single cells. So very briefly, so originally, Dr. Michael Laflamme was like the first to show that you can actually use stem cell-derived cardiomyocytes to repair damaged tissue. And he showed this in the rodent model where he took these differentiated cardiomyocytes and showed that you can have actually engraftment and the survival of the graft post-transplantation in uninjured heart. Thereafter, he created a pro-survival cocktail that would allow the transplanted cells to survive better. These cells, whenever you inject them, they die most of the time because of the hypoxic environment of the infarct scar. At the same time, if these cells are not adhered to the infarct scar, to the tissue, they go through this thing called anoikis because of lack of detachment and they die. So he created this post-survival cocktail that will enhance the engraftment of the transplanted cardiomyocyte into the infarct scar. And anytime you do these transplantations, you really have to keep in mind the model that you're using. So for example, whenever the group did the transplantation of these stem cell-derived cardiomyocytes in the rodent models, they saw one-to-one coupling. And what that means is that the graft of the cells that has been transplanted beats in synchrony with the host. However, in the rodent model, the heart beats about 300 beats per minute versus the human stem cell-derived cardiomyocytes, they beat at a rate of like 80 to 100 beats per minute. So really, there was no way to because the rodent model has such a high beating rate, then it masks any irregular beating rates that you see because of the graft. So because of that, you see one-to-one coupling in rodents, but then when you do this experiments in monkeys and more clinically relevant pig models, you see the appearance of these transient ventricular tachyarrhythmias that are lethal to clinical translation. So one of the recent studies done in Michael Laflamme's lab is that they showed that if you transplant these stem cell-derived cardiomyocytes into infarcted pig hearts, you can see proper engraftment. As you can see in here, you see like nice graft forming. You see a lot of like the development, and you see the sarcomeric proteins actually becoming more aligned. However, With these animals, you see this within 3 to 4 days post-transplantation, you see the appearance of these ventricular tachycardias that become so lethal that you have to sacrifice the animal. So, for example, the heart rate of a pig is about like 80 beats per minute, which is similar to the average human heart rate. With respect to these arrhythmias, sometimes you can go up to 300 beats per minute, which they reach the human endpoint, and you have to sacrifice them. And there's a lot of strategies that are being used in order to reduce these episodes of ventricular tachycardia, because these are the sole critical limitation in pursuing this life-saving therapy into the clinics. So as I said previously, there's a lot of strategies that people use in order to mitigate these arrhythmias. So you can do pharmaceutical treatments, you can do pro-survival treatments, you can use organoids and sheets, you can mature these stem cells of cardiomyocytes, and what have you. So in here, I kind of like use back and forth like synthetic biology and genome engineering in order to better increase the stress tolerance of these cells. So before diving deeper into some of the work that I did, I wanted to talk about the background. So connexins are these gap junction proteins in the heart that allow the propagation of the signal between the neighboring cardiomyocytes. So throughout the conduction system in the heart, we have different connexins being expressed, and every connexin has its own properties. So faster connexins with faster conductions allow for the beat to go to propagate faster, compared to connexins that are slower allow the propagation of these signals to be in a slower. And you need that fast and slow propagation in order to have a synchronous heartbeat. And in here, I'll be focusing on the left ventricle, because the predominant damage that happened during a myocardial infarction event occurs during in the left ventricle. So connexin 43 is basically like when connexin 43 comes together, 6 of them form a connexon, and then when 2 connexons from the neighboring cells come together, they form this gap junction channel that allows the propagation of ATP, ion channels, signals, microRNAs, amino acids, and glucose between the neighboring cardiomyocytes. And you can have heterotropic, homeotropic, and heterotropic gap junction proteins, which means that you can have them all connexin 43, all connexin 43, connexin 45, connexin, the different combinations of the different connexins. So, and depending on the stress condition, you can have, and the type of cell, and the type of heart cell, then you can have a different combination of these connexins. So again, you can see very briefly here, like this is where they're located. So they allow the propagation of ions and signals between the neighboring cardiomyocyte. And if you delete or disrupt this gap junction communication, then you disrupt the propagation of signals, of the synchronous signals, therefore causing arrhythmogenic behavior. And we have preliminary evidence to show that because the graft acts as an ectopic pacemaker, driving the appearance of these arrhythmias, there is a disruption in the gap junction communication. So going back to the central dogma of biology, I wanted to see what is exactly happening. So with connexin 43 is a very important protein. So the C terminus of it is subjected to several different post-translational modifications that allow it to not only function properly, but allow it to basically regulate itself under different conditions of, for example, ischemia, nutrient deprivation, cold, and what have you, in order to regulate the communication between the cells. Because keep in mind that not only are these connexins are important in regulating healthy signals, but overactivity of these connections, if they're open during times of stress, then they can also allow the propagation of damaged molecules from one healthy cell to the other. So because of that, the cell has to basically use different mechanisms in order to regulate the activity. So in here, our previous collaborator has shown that there's several post-translational modification sites on connexin 43 that have been differentially regulated during ischemia. So we see that casein kinase 1 Under normal conditions, connexins are phosphorylated. However, when you subject them to ischemia, this phosphorylation event is actually disappears. And because this phosphorylation event disappears, we have lack of proper connexin 43 activity, and we have increased in internalization, which we believe that was one cause of the arrhythmogenic, the one cause that causes one potential cause of arrhythmia. So, and we have like our we have a study in literature done on mice that where they took basically connexin 43 and they did a phosphomimetic and a dephosphomimetic mutation. So they changed those three serine residues either to alanine to mimic dephosphorylation or to glutamic acid to mimic phosphorylation in order to see what that stability, like what that does to the stability of connexin 43 and how it affects gap junction communication. And what they showed that is that once you have the phosphomimetic mutation, the S3E, then you have more resistance to ventricular tachycardia. So if you induce external stimulation, if you do bursting, then it reduces the episodes of arrhythmic behavior, and it also improves conduction velocity under ischemic conditions. So because of that, we hypothesize that when we change the serine residues that are prone to phosphorylation and dephosphorylation during stress in these embryonic stem cells with phosphomimetic mutations, then we can reduce the incidence of arrhythmia that occurs under ischemic conditions. So very briefly, and this study was published this year, is that we took these undifferentiated cells and we used CRISPR-Cas9 technology in order to change the three serine residues here to phosphomimetic mutations, so that to fool the cell to believe that these cells are always being phosphorylated. So, and then we did Sanger sequencing to confirm this mutation. And it was confirmed that we indeed do have these mutations without any off-target effects. And we did the karyotyping, it did not affect karyotyping. And then we did pluripotency testing to make sure that these cells remain in their pluripotent state. And indeed, they do. So this mutation did not affect the development of the undifferentiated cells. So then we use our cytokine-mediated protocol in order to see can we differentiate these cells into cardiomyocytes? And indeed we could. We could induce mesoderm formation, as you can see by the flow cytometry data here marked by CD56 and CD140A. And then when we keep them to day 20, we can see the cardiac markers as well as the ventricular marker MLC2V being expressed. So for this, we already know that these cells can indeed differentiate into ventricular myocytes, so we can use them for therapy. Then we tried to, let me see if the video works. Then we took, but it was interesting because whenever we saw these cells, their beating was already different, which we weren't expecting. So as you can see, the wild types are beating very synchronously versus these mutants are beating a lot faster compared to their wild type counterparts. And the beating is like very irregular. So that prompted us to like investigate further to see why do we see these changes in the ventricular myocyte, in the stem cell derived cardiomyocyte, but not in compared to what we see in literature. So in here, I use immunostaining on these monolayers to see the localization of connexin 43 under normal and ischemic conditions in both wild type and the S3E mutants. We see that under normoxic conditions, we have the localization of connexin 43 in the periphery of the cell. And then, however, these mutants, even at baseline, without exposure to ischemia, they have lower levels of connexin 43. And it gets even worse when you expose these cells to hypoxic conditions. We see that in the wild type, there's a drastic reduction in connexin 43, and in the CK1 mutants, it's even more reduced. And you can see that with the quantification here, which this was a little bit like confusing for us because we didn't know why this was happening. So then we use optical mapping in order to measure the electrophysiology of these cells. So we took these cells, these monolayers at normoxic and ischemic conditions from the wild type and the mutants, and then we paste them at 1 hertz in order to measure the different properties. And we see that even at baseline, the spontaneous beating rate of the mutants is a lot higher compared to the wild type. However, when we expose them to ischemic conditions, this difference is basically normalized. And this could be because wild type cells lose more connexins during ischemic conditions. So when you lose connexins, your spontaneous contractile activity increases. So it could be because these wild type cells lost more connexin, then they became similar to the mutants. We saw there was no difference in ABT90, but when we measured the conduction velocity, the speed at which electrical signal travels from one side to another, we saw that there was like a drastic downregulation under ischemic conditions between the same phenotypes. So the wild type had a reduction in conduction velocity under normoxic and ischemic conditions, as well as the mutant. But I was also interested in measuring the calcium propagation in this. So in here, like again, we took these monolayers. This time we just stayed at we stayed at normoxic conditions, because right now I was just interested in seeing the mechanisms of how these mutation causes irregular beating rates. So as you can see here, this is just like a monolayer. With the wild type, it's beating very uniformly, right? But then when you see the calcium propagation rate in the mutants, you see that you have these alternans that happen that we think are probably caused because of lack of adequate calcium clear up. So we have the calcium burst, and before it gets cleared up, we have another contraction happening. But interestingly, these alternans disappear once we paste these cardiomyocytes at 1 hertz. So right now I want to see, okay, we have all of these changes. What next? What is the mechanism that regulates connexin 4-3 expression and membrane localization in the mutants? So I measured the transcript levels of some of the ion channels, sarcomeric proteins, and excitation contraction coupling targets at normothermic levels between the wild type and the S3E mutants. And to my surprise, I saw the upregulation of several of these genes. Now we see HCN1 and HCN2, as well as HCN4, which is responsible for the phoneocrine, to be significantly upregulated in the mutant compared to the wild type, which kind of explains the increased in spontaneous activity. And we also see the upregulation of triadin in here, which when you first look at it, it seems that these cells are more mature, they seem to be better, but then once you measure the protein levels, it's actually downregulated. But what's interesting in here is that we know that the protein levels and localization was decreased even at normothermic levels in these mutants compared to the wild type. But when we measure these transcript levels, the levels are actually a lot higher compared to the wild type, although it did not reach a statistical significance, but there was an increasing trend. So going back to the central dogma of biology, or what I learned during my graduate studies and during the previous CHRIB paper, is that I wanted to see, is there any post-translational regulation that's actually affecting the activity of these mRNAs? And lo and behold, when I measured some of the microRNAs that are known based on literature review to regulate connexin 43, I see an increase in several of these microRNAs, including microRNA 13P. At the same time, there was another study that came out from Walt's group, where they looked at the activity of miR1A compared to connexin 43 expression. So if you have an upregulation of microRNA 1, you see a reduction in connexin levels, and if you inhibit this mRNA, then you see an increase in connexin 43. So this already kind of made me think that, okay, even though we do see an increase in transcript levels, the cell, for whatever reason, believes that it's under stress, so it doesn't upregulate the corresponding the protein levels of the connexin 43. And I wanted to go further to see whether having these phosphomimetic mutation, whether it causes the cell to believe that there's other regulations that need to be done on the cells. So when I used Western blotting in order to detect the phosphorylation levels of both total connexin 4-3 as well as the MAPK mediated phosphorylation, I saw that there was a significant increase in the MAPK mediated phosphorylation in the mutant compared to the wild type. And MAPK mediated phosphorylation impairs membrane transport and increases connexin 4-3 localization and degradation. So even though our mutation was supposed to be stabilizing this connexin 4-3 into the membrane, it actually increased its internalization by promoting MAPKs to phosphorylate alternative sites that are known to cause protein internalization. And at the same time, so unfortunately, so we published this study. It was just in vitro, and because it was like already pro-arrhythmic, there was no point of going in vivo. But I was really interested in studying different mechanisms of different tools, basically, to study the health of the heart as well as the downstream activity. So with that being said, I started using these cells that basically have the ASAP reporter in them. And ASAP1 is just like a voltage sensitive reporter that's genetically engineered inside these cardiomyocytes. And if you can see, why is this important is that when you genetically engineer this voltage sensitive reporter, and there's also GCAMs, which is a calcium sensitive reporter, is that it allows you to see the electrical activity of the transplanted cells compared to the host when we take the heart when we retrieve the heart to see the graft activity ex vivo. So